In this video, I want to talk about antibodies in the context of the essentials of adaptive immunity. So, this is a big topic. Antibodies are newsworthy. I've read so many articles asking, with this new variant of SARS-CoV-2 virus, the Omicron variant XBB, will our antibodies be able to handle it? Well, that is a great question, and that is what I want you all to be able to think about. But we have to really start with what are antibodies and what do they do? And so there is this slide, which is great. It says we have lots of functions of antibodies. They can prevent virions from attaching. They can opsonize bacteria. That's awesome. But what are they? Well, they are proteins with hypervariable region that makes an antigen binding site or an epitope binding site. And then they have another non-variable, non-randomly generated region. And this is what other cells can recognize. And we'll get into all of that. So let's draw an antibody, because it just doesn't work to show you all a picture in a video. Basically, what I'm going to draw is a, what are we going to call it? I'm going to draw a conceptual diagram of an antibody. So it starts with two identical proteins. And what's cool about these proteins is that on one end, they are the same as every other version of them, but then they have hypervariable regions. And these went through the kind of random DNA scrambling that lymphocytes go through. In fact, this is exactly what gets scrambled in a B cell early in its development. So this is the part of a B cell that gets that random scrambling. And if you go back, you might think, well, wait, didn't you say it's the B cell receptor? And I say to you, the B cell receptor and the antibody are structurally the same, except for small minor differences. But basically, they are the same thing. So I draw this with two colors because there are really multiple regions that all get randomly generated in order to maximize how weird and random the antigen binding site is. So, I'm going to keep drawing, and I'll talk about where we're at in a minute. So there are two more identical proteins that have the same part in every version of them, and then have the randomly generated parts. So I'm bad at colors, but another randomly generated part, which is going to be identical on this one. And then what about a different color? What about a color I haven't already used? Yeah. And then yet another randomly generated part. Cool. So what we have drawn is an antibody. And I'm going to label parts. So this colorful part is the antigen binding site, which is more appropriately thought of as an epitope binding site. But the difference is you really think of it as an antigen binding site when you're thinking about what antibodies are doing, and you think about an epitope binding site when you think about how they do it and how an immune response happens. So it kind of breaks my brain a little bit to try to decide, should I call them antigens or epitope binding sites? But either way, those are different. They're different. Antigens are different from epitopes. Epitopes are the small fragment of an antigen that actually gets recognized by the receptors. But you could see this either way. So let's keep labeling. This is also called the hypervariable region. And the antigen binding site is made of hypervariable region DNA. So by hypervariable, what that means is that if you compare a bunch of antibodies, they're all going to have different antigen binding sites, but the rest of them is all going to be identical. So part of them is going to be this, what is called the heavy chain. It's called heavy because it's a longer chain of amino acids than the light chain. And so this heavy chain is identical to this one. This heavy chain is identical to this one. So an antibody is going to really have two different proteins, but two copies of each of them. There's one more part of this antibody that we need to label, and I'm going to draw a dotted thing around it and call it the FC fragment, or the FC part. And what this is a constant region. It's not hypervariable. It is constant. It is very predictable, and it is the same in every antibody. And so this is something the rest of the immune system can look for. And it is going to be essential to how antibodies work. So they have two very important parts, the antigen binding site and the FC fragment. And that's what we need to remember. And that is explained in these slides. I just would rather write it out. Partly because the slides are set up so that if you go through them in order, it all builds on itself, but we're not quite going in that order. So it is what it is. So what do antibodies do? Here's really what they do. They stick to stuff. That's all they do. Their binding sites bind to stuff. So they attach to bacteria and viruses, and they opsonize them. Remember, opsonizing something makes it more delicious to macrophages, neutrophils, and dendritic cells, but mostly macrophages. Something that has been opsonized is more likely to go through phagocytosis. So they mark objects to be eaten. They can also obstruct binding of they can obstruct binding of a virion to a cell receptor, and that will make more sense when we look at viruses. But they can basically, by coating the outside of a virus particle, they prevent it from interacting with receptors on the surface of cells, and that stops it from getting into the cell. So antibodies can actually prevent a viral infection. The virus can get in you, but it can't replicate if you already have good antibodies against it. They can also stick cells together and make them unable to move through tissues. So some of those things are drawn here. This is all drawn at different scales. So ignore the scale in all of this stuff because most of it is wrong. So opsonization, for example, is one thing that antibodies can do. They coat it, and suddenly a macrophage notices this FC fragment, this FC fragment, this FC fragment, and says, oh, there must be an antibody on this. I'm going to eat it. But antibodies are much, much smaller than bacteria. Like, if you draw bacteria this big, you can't see individual antibodies on that scale. What are you going to do? Agglutination is the process of sticking them all together, and you can actually make giant networks of cells that are stuck together by antibodies because antibodies have two binding sites. If you get this, the individual cells can't swim through a tissue because they're stuck to other cells. And then this viral inhibition is where they coat the outside of a virus particle and don't let it get into cells. But then also, antibodies protect us from toxins like the tetanus toxin or the diphtheria tracheal toxin. They do that by preventing activity, first of all. And so if you have a protein toxin that is a squiggly chain of amino acids that has a very specific shape, and if that protein toxin works by binding to a human receptor and screwing up the nervous system, like the tetanus toxin, well, if you coat it with antibodies, which if they were to scale, they would be the same size as this protein, but you put an antibody on it, suddenly it is now much bigger and it has the wrong shape, and it can no longer interact with the receptor that it would normally interact with. So an antibody just gets in the way of binding of this protein to a receptor. And then eventually enough antibodies stick to this protein, and then with their other binding site stick to another copy of it. They make big networks of it and make it precipitate, fall out of solution, and become a big object that a macrophage can eat. That's what antibodies do, and I will definitely ask you for a list of that on the exam. If you go back to the last video, lymphocyte development, that whole thing of lymphocyte development, all those steps, all those stages, memorize that. I'll ask you for that on the exam. List of effector cells, 4 different effector cells, and what they do, that will be on the exam. I will expect you to be able to write that. And then what is a lymphocyte? I expect you to be able to blow your friends' minds by telling them about lymphocytes. And if that doesn't make sense, it means you haven't thought it through yet, because once you understand a lymphocyte, suddenly nothing makes sense anymore. If you go back and look at the function of a cytotoxic T cell, it's supposed to kill abnormal host cells. So it kills your own cells if they're abnormal. But it's a T cell. It only has one T cell receptor. How can it possibly tell your cells are abnormal? And in a very real way it can't, and in a very real way it does. So stay tuned for the immune system in Unit 3. We put it all together. It'll make sense. And I look forward to that. So I'll see you sometime.